Hey Team Bio, welcome to your screencast on protein synthesis and secretion. So this might be a bit of a bold statement to say, but I would think that understanding protein synthesis, how proteins are made, and then how they're exported out of cells is one of the most important discoveries of the 20th century and has created the field of genetics and biotechnology. Understanding how this process works is the key to manipulating living systems and to hacking evolution, which is a pretty amazing power. So, I don't know. It's important stuff. And if you could take one thing away from biology, I would hope it would be understanding how DNA encodes the information that then becomes traits and characteristics. And that's what this screencast is all about. So here we go. Okay, so today's screencast is actually going to be broken down into two parts. We're going to start out with protein synthesis, and then the next screencast is going to be about secretion. So synthesis is how proteins are made, and then secretion is how they are moved around the cell or exported out of the cell. Okay, so synthesis. Protein synthesis is what we call the central dogma of biology. It's the flow of information from DNA to RNA to proteins. And proteins give living organisms their traits and behaviors. Okay. So we're going to trace the path of the flow of information and material from the DNA all the way to the protein today. So this all starts out inside of a cell. What you're not seeing here is the exterior cell membrane. So here in pink, we have the nucleus, and here in blue, we have the cytoplasm. And outside of both of these things, holding all of this stuff in place would be a cell membrane that is not shown in this image, but know that it's you can infer it. Okay, so we're going to start out inside the nucleus with the DNA. The DNA, as you know, is a double-stranded molecule that forms a helix. In the DNA, adenine always binds to thymine, and cytosine always bonds to guanine. So if we know one strand of DNA, we automatically can infer the other from this complementary base pairing system. It means if these two strands are separated from one another, we can build two exact replicas of this molecule from either side, which is going to be important later on when we talk about how cells copy themselves and divide into two. Okay, so this whole process begins in the nucleus with the DNA, and it's a process called transcription. Transcription is when you're doing transcription, for example. You're hearing something in one form, which is the form of a video, and then you are transcribing it into a new medium, which is the pen and paper. Or I guess, is that a bad description? Transcription is basically you're taking something. Okay, we have DNA. These are composed of nucleotides, and it is an example of a nucleic acid. Transcription is the process of taking that nucleic acid, DNA, and turning it into a new type of nucleic acid, which is RNA. RNA is very similar in its structure to the DNA, except one big difference is that it is only composed of a single strand, as opposed to the double-stranded DNA, and it has slightly different base pairs. It has A, U, C, and G. The way that the DNA is copied and turned into RNA is that these two strands split apart, and we use this complementary base pairing with, in this case, A to U, and then T to A, C to G, and G to C, to build an RNA from the DNA template. If that doesn't totally make sense, don't worry. More on that in the spring term. But so basically, transcription is the process of taking the DNA, splitting it apart, synthesizing an RNA, and then pasting the DNA back together. So now we have a copy of the information that was in the DNA in the form of RNA, and the DNA is unchanged. And this type of RNA is a special type of RNA because it's going to become a protein. We call it mRNA or messenger RNA because it's encoding a message. So now this mRNA leaves the nucleus through a little gap in the nuclear membrane called a nuclear pore. And it exits the nuclear pore and ends up in the cytoplasm. So this is our same mRNA, except now it is no longer in the nucleus. It's moved itself into the cytoplasm. Here is where the business of translation is going to take place. Translation is where you take an RNA message and from it you build a protein. And I guess what I was trying to convey earlier, which maybe I didn't very well, transcription is going from one form of nucleic acid to another form of nucleic acid. So you're transcribing but not changing. So you're taking the English language written and you're turning it to the English language oral, out loud. Translation is going from nucleic acid to a totally different organic molecule, protein. And the intermediary between the RNA message and the final protein product is the ribosome. So the ribosome here is shown in blue. So a ribosome is an enzyme. It's actually a ribozyme. It's an RNA and protein complex, but it's mostly RNA. I guess you don't really need to know that, but it's true. But it has enzymatic functionality, meaning that it does something. And the thing that it does is it slides along an RNA message and it reads the message. And then it takes amino acids that are hanging out. That's too big. Let's try 6. There are a bunch of amino acids that are just free floating in the cytoplasm. And so the ribosome grabs these amino acids, or it grabs a specific amino acid. It's going to read the mRNA message, and then it's going to grab whatever amino acid is indicated in the message, and it's going to add it to this side of the growing polypeptide chain. So this here is a polypeptide. Remember, the mRNA message, it's a sequence of A's, U's, C's, and G's. I don't know why I did a dash here. And the sequence of the A's, U's, C's, and G's is important. It encodes information telling the ribosome which specific amino acid to pick out of the cytoplasm and add to the growing chain. So remember, there are 20 different types of amino acids to choose from, and the mRNA message is going to tell the ribosome in which order the amino acid should go to create the polypeptide. So remember, this polypeptide is a primary structure protein. And in order to become a functional protein, it needs to be folded into secondary and tertiary structure. And that's a bit about what's going to be happening in the screencast tomorrow, where we're going to talk about this process, how the protein becomes a functional protein by being folded, and then how that protein is moved around the cell and sent to where it needs to go. Okay, that's all.